So welcome to the course of Introduction to Proteomics. So this is the week one live session. So what we will do in this 8 week. So since you know that this is a 8 week course, so what we will do is that in this span of 8 weeks, we will be going through different theories, concepts that you have already are studying in the through the videos. Then we will do some problems, and also we will try to do some hands-on, which will be extra to whatever is being there in the syllabus as per the time permits. So before starting today's session, so first of all, if you all can tell me briefly that what are your I'll come to your doubts. So before that, if you all can tell me briefly what is your idea about proteomics, and after going through the first lecture, what do you feel proteomics is about? Anyone would like to say? And then we will go on with your doubts, and then we will look at the different revisit the different concepts, and we will try to make it interactive because in your video, I think you are listening, and then you are writing in the query, but here you are getting an opportunity to interact directly. So it would be good. So if anyone can tell that if there was any idea beforehand regarding proteomics, or it's first time you are doing, and what was your idea, uh, and what is the change in any idea, or you are to the same, and what was your main idea behind taking this course? Anyone if would like to comment? So what do you mean by this term proteomics? Anyone? So shall I call by name? Okay, so if I give my introduction, so that would make everything clear. So I am Ankit, who is currently in final year of PhD, MTech PhD dual degree from IIT Bombay. So I am a PMRF fellow, and I am taking this. So you can also, if you want, you can also introduce and interact. So for example, for instance, if you can tell about what was your idea behind proteomics, taking this, and what do you think of the term proteomics? Sorry, if you come again, study of protein folding, protein structure. Okay, I think Pooja has raised her hand. Yes, if you can go through study of proteins. Okay, study of proteins. So if we break down the term, yes, Sohamita, I guess. Yes. Mhm. So what is the meaning of proteome? The set of proteins. Okay, so the term, if I say, if anyone would like to try, anyone else would like to try? Anyone? Mary or Pratiksha, Priya, anyone? Shiva, Tanishka. Okay, so you have got almost, so proteome. So the term omics Yes. Anyone was telling? So if we say that omics, it comes from om, right? So that is means all. So proteome refers to the set of proteins that each and every protein that is present in an individual, in a cell, uh, in an individual, in an organ, in an organism. So you can say organelle proteome, cellular proteome. Okay, so from where the term comes of proteomics. So here in this course, you are learning about some basic protein science. I think that is covered in this week one. And next, to study about the proteins, all proteins. So for that, we need to understand, if we try to understand the protein expression of the cell, which we will see, then we need to do many techniques from where we can identify and understand the expression level of the proteins and then understand and correlate in the disease state. So this is exactly the idea behind proteomics. Okay. So which is the entire set of studying of the entire set of proteins, not individual proteins or a subset of proteins, rather the entire proteins. And then in this course, we will be going through that how the field has evolved, how different techniques have evolved, and how it is still rapidly evolving and progressing for understanding of the proteins at a different level, at different states, PTMs, all these comes under proteins, proteomics. Okay. So, but before I going to that, I have also wanted to revisit some concepts. Before that, would anyone would like to tell that by going through this week's lecture or videos, have you found any doubt, or do you have any doubt that I need to discuss it specifically before going into the discussion? Do you have all any pressing doubt or query regarding the course or regarding that particular question, particular concept, particular thing? No? Yes. So, Amita, yeah. Yes, guanidinium chloride can also be used. I will come to that concept as well in my to the presentation. I will come to, yes, guanidinium chloride is also used. That is also a denaturing agent. Yes. Okay, so I think then we can go step by step, go each topic by topic, and if you find out anything difficulty while you were going through the videos and want to clarify, then definitely you can uh, stop me and ask. And as I said that today, since in this week there was not much possibility of having hands-on sessions, but as we go along with the course, we will be having hands-on here in the sessions as well. Okay, so that whatever is being taught in the lectures and whatever assignments you are doing, along with that, you get some extra exposure as well. Okay, so I will now go to the presentation, and in each topic, you are free to ask anytime any query you have. Might be it will be difficult for me to follow in the chat because in the full screen version, uh, I cannot see. Yes, Nisha, I guess. Okay, if it was by mistake. So in the chat, it becomes difficult to follow because in the presentation mode, generally, I am not able to see the screen. But definitely, you can stop, interrupt, and unmute and ask, and feel free to ask and make it as much interactive as possible. You are free to ask any question. Okay. Yeah. So I will just start with some of the concept. Yeah, and if I ask also questions, so if you please feel free to respond, and the way the more interactive we will make, the more we will learn it together. Okay, so I'm requesting everyone to just be interactive and be answerable to whatever whichever answers you know, as well as whichever you don't know, and you have any doubt, like just now Swamita did. So everyone is encouraged to do the same. Okay. So we are going basic, starting from the basic scratch, basic biochemistry, the introduction to amino acids. right? So I think all of you are well aware about the concept of amino acids. Is there anyone who doesn't know in depth about amino acids? I guess no. So it is the building blocks of proteins, and we know that the amino acids are different 20 types of amino acids. Most popular ones, as well as the 21st, 22nd amino acids are also being identified. And in course of time, it might be, we can see that there are different types of amino acids, right? So firstly, there are a major thing that comes into play or action is that the uh, what is the so just a minute, I'll check. So the major thing that I wanted to say is that there are different R groups that makes it diverse, as you know. So there will be aliphatic R groups, uncharged R groups, positively charged R groups, negatively charged R groups, aromatic R groups. So all these makes the backbone is same with the NH2 and the amine and the COH carboxyl bond, carboxyl ion, but the whatever the change in the type of the R chain branch chain or branched, it can be positive, it can be negatively charged. So all this leads to the different forms of amino acids, right? So this is what from there the complexity starts, and from where the field of the concept where the proteins build up, which makes up all our muscles, tissues, organs, and then finally the diversity, the whatever changes that is visible to each one of us is visible because of the changes in these proteins. So you know that in the genomics, after you do, if you do a gene sequencing between me, you, any animal that you take a chimpanzee or a monkey, the genetic sequence or the gene sequence will be, or the DNA sequence will be roughly 99% similar. But still, we all are very much different. And the reason for it is nothing but only the proteins, the way amino acids are done or the way amino acids are developed. The proteins are there, different forms of proteins and performing different structure and function. Then there comes definitely the concept of the alternative splicing and all, where different proteins are formed due to uh, make up different structures, different functions. So all these are very much required to for the field of so in this, to make the things diverse and each and every individual to be different, performing different functions. So all these are very much crucial. So now if we move ahead, so these are basically the amino acid structures. So different structures. So here if we can say that the glycine, which is very small and have only H. So the most simplest form of amino acid is glycine. Of course, you all must be aware of it, which doesn't contain any in the only H in the fourth uh, chain. Then comes CH3 alanine, which is again just the complex version of glycine, which contains CH3. Then we have the serine, threonine, cysteine of the nucleophilic. Then you have the hydrophobic ones, which is the branch chain amino acids, valine, leucine, isoleucine, methionine, proline. Then you have the aromatic amino acids like phenylalanine, tyrosine, tryptophan, acidic amino acids like aspartic acid, glutamic acid, then asparagine, glutamine, and the basic like histidine, lysine, arginine, among which the aromatic amino acids and proline play a crucial role because of its bulky nature, and it imparts a different functional and structural properties to a particular protein. So specifically, proline is of great interest to study. So now in the BL-Lambert's law, before going on, so how many of you are aware about BL-Lambert's law? So who will be telling about BL-Lambert's law? So you all are studying in colleges, right? So there you are familiar with it. So how many of you are familiar and have heard about real numbers law? Please raise your hand. Okay, one Hari. And the others are not aware about real numbers law? You all have Puja, yes. Others? Yeah, yes. So, so who would like to say? Puja, you can go, or Priya. What is real numbers law? The absorbance is directly proportional to concentration and yes, and the length of the pathway of the light, right? So yeah, so before going into that concept of what told, so before that we need to have an understanding what is the incidence, what is the transmittance, what is the absorbance. So what is the meaning of the word absorbance? If I say, if we as you said that absorbance is directly proportional to the concentration and length, so the application lies is that in that absorbance is directly proportional to concentration, so that we can measure the unknown concentration of a particular solution. So that's why Beer-Lambert law is used, and I think you all have done an experiment where you have used and absorbance and measured the absorbance and tried to find out the concentration, unknown concentration of a particular sample, which we don't know. So from a standard graph. So if we know the absorbance of a standard concentrations of a graph of a solution, then from a graph, if we make a particular equation and if we can draw derive an equation, then from for that an unknown concentration can easily be calculated. But before going into that, so what is from where the term absorbance is coming? So firstly is the transmittance. So in this particular, for instance, in this particular cuvette, if we have the solution and if we are incidenting a light, then and it will be finally after getting absorbed, diffracted, refracted, all these things, it will be transmitted, right? So this comes the concept of transmittance, which is the transmitted light upon the incident light. T equal to I by I naught. That what was the initial incident light provided, which was and in the numerator be the what is the transmitted light. So the percentage is T% will be I by I naught into 100. Now absorbance is the negative logarithmic of transmittance. Okay, so what is absorbance? Absorbance is nothing but the negative logarithmic of the transmittance or log 10 I0 by I. So zero absorbance corresponds to 100% transmittance. So zero absorbance corresponds to our 100% transmittance, and vice versa is that 100% absorbance means there will be no transmittance. It will be finally fully absorbed, right? So as you mentioned, A equals to epsilon Cl. So this epsilon is the molar absorption coefficient, and the major thing that has been used is used extensively in our biological studies and research is to find the unknown concentration for the absorbance. Now this is where it helps us in getting particular things. So you can see that for 38 micromolar, there will be a certain absorbance, and for a 152 micromolar, there will be a certain state of absorbance. So absorbance increases quite cold times when there is an increase in concentration. So in this way, if we can measure, if we know, for example, 5 or 6 data points, we know that this concentration, this is the known concentration. We made a standard curve. We get to measure the absorbance for each of these 5 and 6 particular concentrations absorbance, then the seventh concentration, that would be the unknown concentration, we can derive from this straight line, from this equation. So, sorry, so from this equation, we can get to measure the absorbance. Okay, so this is a standard curve we can say. And from here, for the seventh unknown concentration, we can easily, so if we can get the absorbance, so how it is generally done. So I hope that you, every one of you know. So for example, this is an unknown concentration graph where you will be getting. So there are 1, 2, 3, 4, 5, 6 data points. For the seventh data point, the absorbance comes something around here, right? If the absorbance comes here, so we need to extend the line to the place where it is reaching, and then we need to draw the uh, draw the straight line, and whatever concentration it comes, so that is the particular concentration of that unknown sample with the absorbance, for instance 1.3. So for 1.3 absorbance, we will draw the line and draw the and get the concentration. So am I right? So I think all of you know about this, but still I stressed out on the fact. Now tell me that what is the absorbance of tryptophan? So what is the absorbance of tryptophan amino acids or the aromatic amino acid? Mostly it is considered. So you can tell about tryptophan. I think this is 280 nanometer. So all agree with it? Who all agree? Just raise your hand with 280 nanometer. Okay. Okay. Others? Okay. So your voice is not clear, but I could understand that you were saying 270 something. Are you telling like that? Okay. Okay. So this is in which range? 280 nanometers or 270 something like Hadi was mentioning. What is the range? So will you tell me now tryptophan is absorbing at 280 nanometer. Does glycine or alanine, what is their absorbance? Anyone? For glycine's absorbance? Alanine's absorbance, anyone would like to try? Yes. Yes. Basil liver. Yeah, anyone can try. So do you think that the absorbance by tryptophan at 280 nanometer, all the amino acids will have the same absorbance because each and every one is amino acid? What are your take? Let's discuss. For instance, we don't know. What do you feel that, yes, only aromatic amino acids will show. So any particular reason for that? Aromaticity is a property, but what is aromatic ring is there? So then, okay, so aromatic and the aromatic amino acids will have aromatic. Okay, so you have tried. Others can, would they like to extend? Yes. Your I could not hear you. Sorry, Mans. Has anyone able to hear? Then so you can chat, write in chat box now. I can see 5 months, right? Yeah, yeah. So that is the actual thing. So we will see now. I Yeah. So aromatic amino acids absorb at 280 nanometer. Why? Because they have this conjugated ring structure. So what Hari has mentioned it correct, and what Priya was trying to intend to say that yes, it is an aromatic ring which gives it the property, but actual reason is that it has the conjugated pi bonds. Okay, so these pi bonds are are in a delocalized state, are not in a very localized or conformed state. So what happens is that whenever an UV light is passing through it, and if one molecule gets absorbed to it, so the electrons, so what is the basic fundamental? That the fundamental is that the electrons will absorb that, will go to a higher energy. So if that goes to an higher energy and that higher energy is unfavorable, then what will happen is that the bond will break, right? But here what happens is that in the aromatic amino acid, it is in the state of cation pi interaction. Okay, so it's stabilizing electrostatic interaction between a cation and the polarizable pi electron cloud of an aromatic ring. Okay, so this is in a stable cation pi interaction, and each carbon atom will have this p atom over orbital overlapping, and it is forming a conjugated pi orbital system. So this creates the delocalized electron cloud. So since they are delocalized, once they absorb your UV light, they go to the higher energy level, and it helps to make it stable. Means the bonds are stable and they do not break down because they are delocalized, and they can absorb the energy and can go to higher energy phase, and still can would be able to keep the bond intact. Okay, so this helps in stability after the, so delocalized pi electrons are the ones that are responsible. So in the why in the so as Priya was mentioning that aromatic amino acid, because of their aromaticity property, but actually not because of the aromatic rings, they have a conjugated ring structures and have a delocalized pi electrons that whenever after absorption also, they help to, after gaining energy also, that the bond remains intact and they can absorb it. Okay, so it attracts, and so it helps in the stability, and then what we get is an enhanced absorption. So the tryptophan is at what Hari was mentioning is correct, and something 279 or around that. So that is the exact absorption, and all other uh, amino acids do not show this structure or do not show this property because of the absence of these pi electrons or the conjugated ring structures. That's why aromatic amino acids are ones which shows the highest absorbance at 280 nanometers. Okay, so am I clear, or anyone has any question? So here we can see that the tryptophan's absorptions, phenylalanine absorption, and the tyrosine's absorption, which is well, where remains around 280, whereas phenylalanine is in the range of around 260 nanometers. Tryptophan is in around 280 nanometers, around something exact, you can figure it out, and tyrosine also in that range. So this is where our particular amino acids gets absorbed, the absorption of aromatic amino acids. Okay, because of this pi electrons and the delocalized structure, that absorption also does not let it break away and keep it intact. Okay, so any question here? Understood it correctly, right? So why we started with the Beer-Lambert law and absorbance is that because we must understand what does we mean first of all by absorbance. Second is that we are telling and we are stating the fact that absorbance is shown by the aromatic amino acids at 280 nanometer, but we should understand the reason behind it. So why and how the absorption is getting increased, and 280 nanometer only the aromatic amino acids are showing a strong absorbance, whereas other amino acids are not showing the absorbance because of this pi electrons. Okay. So is it clear for everyone? So if you can just raise your hand that it is clear, or uh, does any one of you have any doubt? No, right? Great. So now what we will do, is that we are going through the types of amino acids. I think it's a very common question, and I think all of you know. Just anyone just iterate, and then we can skip and go to the next part. What are the types of amino acids according to the dietary requirements? So essential and non-essential amino acids, right? Essential and non-essential amino acids, right? So what are essential amino acids? So essential amino acids. Yes, yes, Pooja, go ahead. Correct. So essentials are the ones which our body are not able to synthesize and are required by in the diet, whereas non-essential ones are the ones that can be synthesized by our human body from other compounds, that is mostly other precursors, and that we do not require to intake from our diet. So like histidine, isoleucine, leucine, tryptophan, threonine, valine, phenylalanine, all these are very much crucial and that needs to be come through our diet. So that's why we should have protein in our diets which contains these particular amino acids that are not able to be synthesized in our body for our functioning and maintenance of the homeostasis and all the tissue, cellular functions, repair, and nutrient absorption. Whereas non-essential amino acids are those that can be synthesized by the human body from other compounds, and that's why it is not absolutely necessary to take it. But yes, of course, we can have it. Okay, so different metabolic processes, enzyme production, and we know that the amino acids, as I mentioned already, that are the sources of the building blocks and every repair, organs, tissues, muscles. For all these things, it is quite important, right? So now, yeah, sorry. So yeah, so now another concept of chirality of amino acids. Yes. So before going again, so if anyone, what do you mean by chiral? Chiral carbon. Have you heard about the term chiral? You must have. Why if you when different groups are attached. Krutika, is it what you told? So different groups. So not different groups. It should be that the four different groups. Okay. So the term four is quite important here because if you have only H, okay, so then you can change it according anything, but it will be mirror superimposable. So if you have in carbon, you know, every one of us know that carbon has four hands, and here if there are four different forms of groups, that's why glycine has no chiral center, if you remember, right? So there must be four different groups that will only make the carbon atom a chiral one. And that will not be superimposable with the image, right? So now what are, so with this, it's to the configuration of the different isomers. So first of all, if you just keep on noting down, first of all, that the amino acids that we have said that it comes under the varied R groups are the ones that leads to the change and the different forms. Then comes the different kinds of isomers, among which due to the carbon and it has our four different groups attached to a single carbon atom, then it gives us a chiral molecule, which is if we change the orientation of each one of those particular molecules, then it will be its mirror image is not superimposable to each other. So here comes the concept of stereoisomers, which is the special arrangement, and enantiomers, which are the optical isomers and have mirror images that are non-superimposable. So enantiomers are both are same. They have the same molecular formula, function, physical property, everything, but their mirror images are not superimposable. So what does enantiomers do? Enantiomers rotate the plane polarized light in dextrorotatory or levorotatory methods. So L amino acids are predominantly found in proteins, that is, which rotates in the levorotatory methods or the left hand rotation, the plane polarized light. So can anyone of you tell where are the mostly D amino acids found? Any example of D amino acids? We are saying that the in the proteins, yes, D, ribose. No, no, ribose sugar we are not talking about. In the amino acid, D amino acids are prevalent where? Any example you know that D amino acids are present? So, so we all know that L amino acids in humans and proteins, which we are saying that L amino acids are present. So try to find out whether so you can after this class and all, you what you can do is that you can go ahead and find and search that whether bacterial proteins contain L amino acids or D amino acids. Okay. So you can take this task, just you can search after your this class that whether after this live session that whether bacteria proteins contains L amino acids or D amino acids. Okay. So that you can do it. So now what we are going to see is that stereo configuration of amino acids. So one of the amino acids, and I think that is also very well explained in your video regarding the D amino acids and the L amino acids and levorotatory and why dextrorotatory and why the enantiomers are different. But another concept is there about the stereo configuration. That is the absolute configuration that refers to the spatial arrangement of the atoms, right? So here the terms are R and S. R refers to rectus and S refers to sinister. So another which is the absolute or the spatial arrangement comes with the configurations of two things. One is R and another is S. That is rectus and sinister, right? So here how it is determined? It is determined on the basis of the priority rules. So what is that priority rule? That the groups attached to a chiral center are ranked according to their atomic number. Okay, so they are ranked by their atomic number, with the highest atomic number is assigned the highest priority and the lowest atomic number is assigned the lowest priority. Okay, so this is the configuration to determine the R or S. L or D is fine. What is will be the R and S. Okay, so this comes with the priority rules that help us in understanding. So there the highest atomic number is assigned by the highest priority, lowest is assigned by the lowest priority. So to properly orient the molecule, the group with the lowest priority is positioned away from the viewer. So then we need to trace the path from priorities like A, B, C. If the path comes clockwise, then it is R. The chiral center is assigned the R designation, and counterclockwise it is receives the S designation. Okay. So what priority rules comes into picture is that the chiral center different groups that are attached, they are marked as A, B, C, D on the basis of the highest priority, which is having the highest atomic number, and the lowest one with the lowest priority. And then what it is done is that it properly orients the molecule. The group with the highest is first of all taken away, and the path is traced back. And the path, if the path is clockwise, then it is R, and if it is not, then S. Okay. So is it clear for each one of you? The R and S. Have you any doubt regarding it? So L and D is one, but the absolute configuration and the absolute spatial arrangement is given by R and S. Okay. So Dectus and Sinister, and this is done This is determined on the basis of certain priority rules. Okay. So now we can move it forward. So after the arrangement, yeah. So after the arrangement is done, okay. So after the arrangement and all, we have seen that D amino acid, L amino acid, special arrangement of amino acids, etc., etc. So now comes to the point of the ionization of amino acids, right? So I think all of you must have heard about the term Zwitter ions. So now we will look into what is actually this Zwitter ions belong to. And believe me that understanding all these basic structures will only further help us in understanding when we are doing understanding the complex concepts of understanding the proteins. So as you mentioned that understanding the proteins, proteomics, for example, instance, uh, relates to the understanding of the entire set of proteins in human beings, and that we can do through different structures, different techniques. But each and of this technique requires the basic understanding of this chemistry, because then only we would be able to break down the proteins, try to analyze those proteins, try to identify those proteins, and then only able to come into some conclusion of identification as well as quantification. So that's why this particular uh, thing is quite important for us to understand the basic chemistry. Then only we can go ahead and study the complex data sets and analyze complex samples and complex molecules. Okay. So here you can see that this is the alpha carbon that we all are aware about. This is the R group, COH, and the amino group. So because of this unique property of this carboxyl group and the amino group, so this is in always in sometimes it is in the positive state, NH3 plus, sometimes in negative state, CO minus, sometimes it is in positive state and a deprotonated state and the protonated groups, right? So the basically this is measured in terms of pH, right? So this whole concept that this particular amino acid will be in protonated form or deprotonated form, right? It will be in deprotonated form, that is in the acidic state, or in the protonated form, that in the basic state, is determined by the pH. So each, or if it is completely dependent on the solution, it is. So the amino acids are often referred to as Zwitter ions, so contains both acidic and basidic groups that allows the intramolecular acid base reaction. So intramolecular, not intermolecular. So please pay careful attention to the term of intra and inter. When it is inter, it is between two molecules. When it is intra, means it is within the molecule. So intra represents anything within. So intramolecular acid-base reaction, where the acidic proton from the carboxylic acid protonates the amine. So from the amine group gets protonated from the carboxyl group, and the it becomes in the either it can be in the when it happens, then it is in the neutral state or the Zwitter ionic form that you can see in the blue when you have the balanced charge H plus and minus. So this Zwitter ionic property is prevalent at physiological pH in aqueous media, characteristic of living organisms, and in other polar solvents. So that is characteristic of living organisms and in your other polar solvents. At different pH values, amino acids can become slightly ionized, favoring either cationic or ionic forms, right? So different pH levels will uh, uh, different pH levels will act differently, and it will favor either cationic forms or ionic forms. And this pH range at which an amino acid is Zwitter ionic varies depends on its specific pK values. So now you have understood that it depends on the different pH, and it forms different acidic state and the basic state, and it keeps on interchanging. So can any one of you tell an application on which it is there? for an application of this property of this amino acid to separate different proteins or different or any kind of application, if you can talk about using this property, any technique that has been developed using this property. Any one of you? So how many of you have heard about the term gel-based proteomics or SDS page gel? SDS page, how many of you have heard? How many of you have heard the term SDS page? No one? Yeah, Priya, yes. So what is the principle of SDS page? Priya, you can go ahead. Yeah. Based on their molecular weight, right? So that is one dimensional. So have you heard about 2D or two-dimensional gel electrophoresis? Okay. So you are not okay. So who else have heard about two-dimensional? I think SDS page all of you have heard. Is there anyone who does not know SDS page or haven't heard of or hearing it for the first time? Sodium dodecyl sulfate polyacrylamide gel electrophoresis. Is there anyone who is hearing for the first time? I would be happy, happiest to see if to learn that if someone is hearing it for the first time, then it will give us an opportunity to discuss. And so I am hoping that I am assuming that everyone knows. So just raise your hand and say that how many of you have heard about the term 2D gel electrophoresis? Yes, Hari and others. Anyone else have heard about the term? I could see Samiksha or someone raised your hand. So you can go ahead, Samiksha, or just try, give a try. I have given you a hint as well that we are reading about the Zwitter ionic structure and those of which principle, which technique has developed on the basis of this principle. Correct. So that will be the amino acid will be in which state at that point of time? Correct. So the basically it is in the range. So we understand that it is a pH which is the amino acids to be in different state. So on the basis where that particular amino acids particular ionic state will be zero, that the charge will be zero, that it will be balanced, right? In the Zwitter ionic state, it will be balanced. So then there will be no more charge separation or the protein won't migrate to the other part of the charge. Otherwise, what is the case and what is the happening is that when the protein is positively charged, so it will be moving towards the negatively charged uh, particular towards the negatively charged uh, cathode. So this is how protein move or the amino acid moves or the protein particular that moves. But when the charge will become zero, then what will happen is that it will stop moving it further. And in the two-dimensional gel electrophoresis, so that is the property of the protein, but from the concept of the amino acid, we can understand that these they are separated on the basis of the pH gradient is formed where the proteins are separated to some extent where their pi isoelectric point becomes zero, and then further if you have a molecular weight based separation like the SDS page, then they will again further separate. That we will definitely come when we are discussing that in detail, like the gel based proteomics. Okay, so we will have the 2D page, 2D dye, as well as the SDS page, we will discuss it in detail. Okay. But I just gave you a hint and idea about how it forms. So now we have talked all about amino acids, but now we all are in the course of proteomics. So we are in the course of proteomics. And we know that we are aiming that we will be able to understand all the proteins in an organism, be it bacteria, be it virus, be it human being, be it a monkey, chimpanzee, dog, anyone. So we are aiming that we will be knowing that all sets of protein, entire sets of protein present in that particular organism. For that, we have understood the structure of amino acids. So now so the proteins cannot be built without the amino acids joining together and joining by a very bond, that is the peptide bond, right? So anyone, what is the name? What can be the other name of the peptide bond? Anyone, just quickly, what can be the other name of the peptide bond? The bond can also be called as. So what is this reaction called and lead to formation of what bond? What type of bond rather? So two amino acids are joined by what reaction? The reaction is known as condensation reaction, right? So anyone who does not know condensation reaction? So what this bond can also be termed as? Anyone, just give a try. What this bond, the peptide bond can also be termed as what type of bond? This is what type of bond actually? So it is a kind of amide bond, right? So what is so peptide bond is nothing but composed of amino acids. So this is one amino acid, this is one amino acid linked by a peptide bond, by covalent peptide bonds, and it is sequence are determined by tRNA that I think all of you must have read or you can read in any molecular biology books, concepts like the tRNA, which will come, will bring the amino acids together and the process of complete process of translation. So it is a condensation reaction. COH group of one amino acid reacts with NH2 group of the next. And what we will do is that it releases H2O. So here one water molecule is released. That's why it is the term is condensation reaction. So peptide bond is formed with condensation reaction where COH group of one reacts with the NH2 group. So here you can see this is the COH part of the group. This is the NH2 group which reacts and releases H2O, right? So this is the condensation reaction. So N terminus is that first amino acid retains NH2 group. So this is the N terminus. So here you can see, please pay careful attention because this is very, very important to understand when we will be analyzing our data as well in the from the complex data sets, when we will be doing the particular cleavage of certain peptides to generate from the proteins to generate peptides and then run in different modern instruments to get understand, identify which amino acid it is or sequencing of the amino acid and quantification of the amino acids. This concept will be very, very useful. So N terminus is this one, which states that the first amino acid retains the NH2 group and loses the COH group to the to bond with the second amino acid. And in this way, ultimately, when 1, 2, 3, 4, whatever is being determined through tRNA, right? Whatever is being determined through tRNA synthesis, when it will be all the sequence will be covered, then the C terminal will remain free. One place where N terminal will remain free, N terminus will remain free, and the other place the C terminus will be remain free. So that is the last amino acid that is known as the C terminus. So a peptide bond starts from an N terminus and ends at C terminus, right? it starts or begin with N terminus and end at C terminus. So the first amino acid retains the NH2 group and loses COH group to bond with the second amino acid, and the last amino acid retains COH group and loses NH2 group with the previous amino acid. So it has the strong with partial bond, double bond character. Okay. So the major thing, so it will come in detail now to understand the characteristics of peptide bond. So this is very, very crucial. Okay. So the characteristic of peptide bond to understand is very, very crucial. So it is strong with the partial bond character, and it is not broken by heating or high salt concentration. So that we need to understand. So this has a strong, means it is not so we can just by heating or something we can uh, denature or broke the heating or the peptide bonds. So it can be broken by strong acids or bases at elevated temperatures and specific enzymes, that is digestive enzymes. Or you can heard, I don't know if you have heard about the term proteases. There are different proteases that can break this particular bond. So peptide bonds are rigid, planar, stabilizing the protein structure and contain partial positive charges and partial negative charges from the oxygen atoms of carboxyl groups and hydrogen atoms of amino groups. So now just a quick question that if a peptide bond is formed by a condensation reaction, that is H2O is removed and carboxyl group and NH group gets joined, then with what reaction can it also can it be broken down, or the peptide bond can be broken down? Any reaction that is coming into your mind to denature the peptide bond or break down the peptide bond? Yes, Hari. Yes, so hydrolysis is one reaction through which the peptide bonds can be denatured, that is, can be broken down. But in normal condition, it is very, very slow process, and it requires certain kind of heating or enzymes, mostly what we use, kinds of proteases. So one task you can go ahead and take it forward before going into the complex proteomics. When you will be doing, you will be, so I can give you a kind of a spoiler that you will be hearing the term trypsin or proteases. Okay, lyse, chymotrypsin, you will be hearing it, or if you read any manuscript or read any particular book, you will be coming across this term. So one task you can take it forward. So these are obviously not related to your assignments or those things, but obviously for your own interest and own understanding, deeper understanding of the subject, like the trypsin, how it works at, so not at what terminal and not what after amino acid. What how it works, what I mean to say is that what is the exact mechanism of how it works. It's fine that it works after certain and certain amino acids. So if you just Google it, you will find that trypsin cuts after C terminal of this and this amino acid. So that is not my question, or that is not what I meant to understand. What I want you all to find out, that what is the exact mechanism, what is the exact reaction it is involved, and how it is done. Okay. So if you want to go ahead for a better understanding, you can take this question and can try to find out. And in the subsequent weeks, when we will be meeting and when we will be discussing, you can come across and you can we can discuss. Okay. So that is one task you can take it forward. So now in that peptide bond, we have seen, we have said that it is a partial double bond character. I don't know how many of you have given attention, paid attention to that particular term. So it is a partial double bond character, though it is a single uh, covalent bond, but it is, we are saying that it is a partial double bond character. It is coming. So can any one of you tell that the particular, what will be the hybridization state of that peptide bond? Does anyone of you like to try? Anyone, just give a try. So all of you are aware about hybridization, right? So what will be the hybridization state of the peptide bond? According to you, according to what I said, that is the partial double bond character. You have understood, right? COH carboxyl group, NH2 group. Yeah, Hari, I think you can wait a little bit because others can also give a try and then you can tell. Anyone else? I'll come to you, Hari, because but let others also try. Yes, so Amita, SP2 and planar. Okay, others? Do you agree? If you agree, you can raise your hand, and if you disagree, you can just. Okay, others? So who have no idea, you can also say that I don't have any idea. So those who have no idea at all that what hybridization state can be, also can raise your hand. If you don't raise your hand now, then I have to ask each one by name by name. Okay, so Swamita, can why did you told SP2? Can you just explain? Swamita, you are there. So can you explain why did you told SP2? Mm-hmm. Hmm. Hmm. Correct. So I'll explain it in a little bit. Where I'm coming to that question. Yeah. So I will. So I have already asked you the question of the hybridization state. So now let us see that why. What could be the plausible reasons for that? Okay. So the characteristics of the peptide bonds, if we talk about of the peptide bonds, so first of all, it absorbs at 192 to 30 nanometer and susceptibility to UV radiation. Okay. So as she mentioned, it is really true that peptide bonds exhibit resonance with partial sharing of two pairs of electrons between the amide nitrogen and the carboxyl oxygen. The amide nitrogen and the carboxyl oxygen. There is a partial sharing of the two pairs of electrons, and this exhibit the resonance structure, resonating, and that's lead to the having the partial double bond character. So the atoms are in the same plane. So first of all, planar, that is they are in the same plane, and hydrogen of the amide group and are trans to each other. So again, we will be now looking further why they are trans and why what method they are. So it was Pauling and Corey determined that peptide bonds are rigid and flare. So you can see that though they have the single bond, there could have been the rotation, but there is a barrier to the rotation because of this lone pair of electrons being resonating with each other. Sorry, and it somewhat feels that it is a double bond in nature, double bond in structure. So that's why they are not able to rotate. There is a huge barrier to the rotation. So this significant barrier to rotation as like a double bond. So then we can say that can you guess the hybridization state? And you have said that it is in sp2 hybridization. So why? So as correctly mentioned, these are resonance hybrids. So there is a resonating structure. So we just have the partial double bond characteristics. So C and N are each sp2 hybridized. We can see that it is sp2 hybridized and it forms a double bond. So why it is that? So sp2 hybridization, what is the characteristics? So they remains planar in 120 degrees. So you can see the resemblance of the structure with the structure that are general properties of the compounds that are in sp2 hybridization, right? In the 120-degree planar to each other. So one s orbital and two p orbitals mix to form three equivalent sp2 hybrid orbitals, and it is known as the trigonal hybridization, trigonal hybridization. And these are arranged in a trigonal planar geometry with 120-degree angles between them, right? So these are with the 120-degree between them, and this hybrid orbital has 33.33% S character and 66.66% P character. So these molecules with a central atom that is linked to three atoms and sp2 hybridized exhibit a triangular planar shape. Okay. So this is the main reason why this is particularly sp2 hybridized state, and this is the structure that is being resemblance of the peptide bond. Okay. So is there any question or query or any thing here? No, right? Okay. So then we can move to understand that the peptide bond that remains mostly in trans form, right? So the not in the cis form. So again, the peptide bond can be present in cis or the trans. So now why they form the have a planar trans configuration and with minimal rotation. So peptide bonds have a planar and in the trans configuration, which is generally having a very minimal rotation. So NC and CO bonds are the intermediate between our single and double bonds, that is, which is having some 0.132 nanometer bond length. And what is happening is that the imido, um, yeah. So amido tautomerization and bond rigidity prevent the free rotation. So what is tautomerization? Anyone? So we have used this term tautomerization. What is the meaning of that? Anyone would like to say what is tautomerization? Yeah, so basically the moving of the protons, right? The H from one place to another. So this helps to, these are all preventing the free rotation. So you can understand that the structure of the peptide bond is quite very much uh, in the format which is planar, which is rigid and doesn't allow for a free rotation to go. So the shared electrons between nitrogen and oxygen contribute to the bond stability. So you can see that the peptide groups in the planar trans configuration. So to maximize why they are in trans state or trans configuration, because they are to maximize the side chain distance, but the NH group of prolines can either cis or trans. So only the proline some ring can be either in cis or trans, but no, maximum peptide bonds are in the trans configuration only. And these transform are in the more common in proteins. So some proline residues, such as permissive, can exist in either cis or trans configuration. So these cases, we can see that some proline residues are permissive to having either of the configurations, but mostly the peptides remain in trans configuration because of to maximize the side chain distance. First of all, because the side chain, if the side chain distance is minimized, then there will be a steric clashes. Okay, there will be a hindrance to the steric, and there will be the whole structure of the peptide bond would be very much disoriented and disorganized. That's why the side chain distance needs to be maximized, and those peptide bonds are to be remain in the trans configuration. Okay. So now there is a question of the someone asked a question as well, and this is the experiment of Anfinsen experiment regarding the protein folding and how the protein can be denatured. I think this can be answered by any one of you, and then we can look at it. So before, so anyone would like to comment on Anfix's experiment? I think someone asked question also. So just give a brief about Anfinsen's experiment. Anyone? So what were the reagents used? So in the beginning only one has asked the question. I don't remember who was that. But yes, beta mercaptoethanol, right? Beta. So what was the role of urea and what is the role of beta mercaptoethanol? So beta mercaptoethanol breaks the disulfide bond, right? Okay. So what happens when the two reagents are given? Correct. So it is reduced to the primary structure from the folded state. And when the reagents are withdrawn, then what happens? Correct. Correct. So what we can understand is that after the reagents are removed, what we can say that it is retaining its property or state, and which is mostly that we can also say that the protein folding is quite we can understand the protein folding in vitro state. So we will go just see each and every step just by looking at it, we will just brush up. So this is was done on the protein ribonuclease, right? So this is the disulfide linkages of the tertiary structure. So which we will again come in a while. We will look at each and every topic, each and every of the secondary structure and those structures. So here the disulfide linkage is there. So if we have the reducing agent and heating or addition of urea, so we will have the unfolded ribonuclease. So if we just give the reducing agent, so the disulfide bonds would be breaking. So you can see that the disulfide bonds which was present is getting out, and then when we are heating or addition of urea, it is unfolding. So what is happening is that the disulfide bridges in proteins are broken by treatment with beta mercaptoethanol, creating new bonds between sulfurs in two mercaptoethanol molecules. So this is quite a standard procedure. I think those who are aware about the concept of DSP or those, they also know the principle behind using beta ME. So this is included in protein separation. So SDS page to ensure the denaturation. So Anfinsen, what he used is that ribonuclease A to demonstrate that protein folding information is contained in the amino acid sequence. So what he wanted to state from this simple experiment is that the protein folding information is contained in the amino acid sequence. And this complete denaturation required urea plus 2-mercaptoethanol to break disulfide bridges. Okay. So this complete denaturation is required to for complete denaturation using urea and 2-mercaptoethanol. Okay. So ribonuclease A refolded spontaneously and began biological activity. So it was refolded and regained the biological activity upon removal of urea and 2-mercaptoethanol, showing refolding can occur in vitro. Okay, so showing that refolding can occur in vitro. So these so now when if you remove just only 2-mercaptoethanol and not urea, it resulted in only 1% activity recovery due to random disulfide bridge formation, right? So when you are using ribonuclease A and the protein folding information is contained within the primary structure, when you are just removing the disulfide linkages, what it will happen is that it will denature the disulfide linkages, and then if you give urea, it will completely denature. But uh, when you remove urea and that mercaptolithanol both, it will show refolding. But when you are removing two mercaptolithanol but not urea, it will result in only 1% activity recovery due to the random disulfide bridge formation. This suggests that the need for correct conformation to achieve full activity. So what we could learn from these experiments, one was that the structure conformation in the primary amino acid, then is that the complete removal of these two things and need to have a correct conformation to achieve full activity. So the enzyme PDI helps correct incorrect disulfide bonds, allowing proteins to unfold and defold correctly, improving correct protein conformation both in vitro and in vivo. Okay. So now we are moving into the understanding of the structures of protein. Okay. So Atkinson could help us understand that whether proteins structure information is confirmed, and the proper structure is important to understand and to regain biological activity. So now if we talk about protein structures, the protein structure, as you can see here, are in can be said into divided into four types: primary structure, secondary structure, tertiary structure, and quaternary structure. So now after seeing these, who would like to state that what is the basic differences between each of these structure, or what are those structural forms? So in the primary structure, what is the basic structure? Primary structure of amino acids means what do you mean by primary structure of amino acids? So you have already seen in the presentation. So what do you mean by primary structure? And in the Atkinson's experiment also, you could understand that after protein is denatured, you are getting the primary amino acid structure. Yes, go ahead. Yes, Bhaswati, you can go ahead. Yes, Bhaswati, you have raised your hand, right? You can go ahead. Okay, right, good. Yeah, so you have mostly stated the definition of the structure. So primary structure is what? Is just the basic sequence, or that is the sequence that has been what we have already discussed, that it is from the after the translation or the using tRNA, we have got the sequence. So this is contains the basic structural information of amino acids within it, and followed by the secondary structure is the uh, holding between the residues of a primary structure, and then goes to the tertiary structure between the different regions of that particular polypeptide, and finally quaternary structure between the two subunits. So now what we will do is that we will go to understand. So before going into what is a pi and what is psi bond, so anyone just quickly you can tell what is pi and what is psi uh, angles or psi bonds in the proteins in an amino acid primary structure. So what is a phi bond? So what is phi and what is psi? Just quickly, anyone? So you all know, right? Sorry, if hmm. So that is C alpha, that a carbon alpha carbon with the C and alpha carbon to N, right? So that is the psi and pi bonds, right? So this is what led Ramachandran or the Ramachandran to decide on the structure of the proteins, that which particular region are we will be able to rotate, will be able to get, and which will be the torsions and those angles, right? So the N C alpha and the C alpha C bonds in a polypeptide which are free to rotate with the rotations represented by the torsion angles phi and psi. So you just try to remember and understand that we have said that the peptide bonds that we are seeing is quite rigid and have a huge amount of barriers to rotation and all these things. Now followed by that, what we are saying that the NC alpha, so now the C alpha C and the pi and the psi angles, that how they are going to rotate in the polypeptide, that is determined by the Ramachandran plot. So that what how it has been determined, that it has been determined to use computer models to vary phi and psi angles systematically to identify the stable conformations and avoiding statically disallowed structures where atomic spheres. How that was determined, that theoretically disallowed structures or atomic spheres. So the based on van der Waals radii would collide. So on the basis of the van der Waals radius, that where it was going to collide, it was decided and it would be taken forward. So whichever regions were allowed, right? So what are white areas in a Ramachandran plot? So in the white areas of the Ramachandran plot are that it is sterically disallowed conformations where atoms come closer than their van der Waals radii, except for glycine. Except for glycine, the atoms come closer and there will be sterically disallowed conformations, right? Sterically disallowed conformations. So this is, so you can see in the fourth quadrant, there is no place where it is allowed region. Okay, so just see, check here. So this white space, whatever white space are there, all are sterically disallowed because their conformations, their atoms cannot come closer. So only these regions are coming closer. So red areas are no steric clashes representing alpha helical and beta sheet conformations. Okay. A yellow area slightly shorter van was radii. So this is only the red regions that represents the no steric clashes and alpha helical and beta sheet conformations can be there. Yellow areas are what? Slightly shorter van was radii show an additional region for the left-handed alpha helix. So this is the left-handed for left-handed alpha helix. This is right-handed alpha helix region, allowed regions. So A amino acids generally don't form extended left-hand helices, but glycine, asparagine, and aspartate can adopt this conformation due to side chain stabilization. So A amino acids do not form this kind of left-handed, extended left-handed alpha helix conformations, but glycine, asparagine, and aspartate can adapt to this conformation due to the side chain stabilization. And glycine lacking a side chain can cut up phi and psi angles in all quadrants of the Ramachandran plot, often found in protein turn regions where other residues would be sterically hindered. So glycine is only one amino acid that is adapt phi and psi angles in all quadrants of the Ramachandran plot, often found in protein turn regions where other residues would be sterically hindered. So if question at all comes in any of the examinations or anywhere, that which particular amino acid is only present in all the four, can be present in all the four quadrants of Ramachandran plot, then please remember that glycine is the only amino acid that can be present in each of the four quadrants of the Ramachandran plot because of its simple nature, and it does not contain any hindered structure or sterically uh, hindered regions, and it is generally found in the protein turns where other amino acids would not be present because of the steric hindrance. Okay, so this is what you can try to understand and remember. So these are the regions which are the disallowed, which are the allowed, and which are extended left-hand helices with have steric clashes, and which does not allow for any regions. Okay. So what is the secondary structure of amino acids? So secondary structure of amino acids can be termed as refers to regular. So we have understood that the primary is just the sequence, but a protein from an instance experiment also you have understood that the to have a biological function, a protein should regain and reform and should have its active structure. Then only the proteins would be able to function it biologically and would be biologically active. Otherwise, it is not possible for the protein with only the particular amino acid sequence to have its complete function, right? So now the next part comes under the process of secondary structure of amino acids. And again, the amino acids or the proteins to be in a particular angle and particular region of the phi psi angle. Again, Ramachandran plot comes into action. Otherwise, there will be steric hindrance and the proteins would go in an unfavorable position, and that residue would make the protein disorder. So what are the two I think you all are aware about it, that the most popular two structures of amino acids are alpha helix and beta strand, are those which are very much crucial and which are very much popular for very much being proteins follow these parts to get it forward. So for example, alpha helix, which is the right-handed spiral conformation. So backbone NH group donates a hydrogen bond to the backbone and CO group to the amino acid, four residues earlier. So what is that meaning? So in the primary amino acid sequence, you have the complete residues. So from I to N, from 1 to N residues. So I and the hydrogen bond develop between the four I plus fourth residue of that particular amino acid, that leads to the particular alpha helix that we will be looking at in detail. Whereas beta strand are the stretches of polypeptide chains, which is 3 to 10 amino acids long in an almost fully extended conformation. And adjacent beta strands from beta sheets that are stabilized by hydrogen bonds. So it includes the beta sheet structure in your silk proteins, which you see that the fibrin and the fibrins, which happens is from the beta sheet that comes into place, and stabilization by hydrogen bonds connected by tight turns and flexible loops. So as what I was mentioning, so this is how the alpha helix loop, right? So if you see, this is the one, so you can follow the number. So what does it mean, the alpha helices, or what is the meaning of the term of alpha helices? That what we are saying. So if we take this particular amino acid as 1, 2, 3, 4, 5. Okay. So what I have said, if you just remember what I just now stressed, is I plus 4. The hydrogen bond interaction between I and I plus 4th residue is what gives you the alpha helix structure. So for that, amino acid is arranged 1, 2, 3, 4, 5, and there is a interaction. So I 1 and 5th. So you can see first and fifth are having one hydrogen bond. Sorry. So this is one hydrogen bond. Please look carefully. If you have any question or any doubt, you can ask. So 1, 2, 3, 4, 5. So 1 and 5 have a bond. 2 and 6 will have a bond. So I and I plus 4. So when it is 2, it is I plus 4 equals to 6. There is a hydrogen bond. Now 3 plus 7. There will be an hydrogen bond. So you can see 3 and 7. Finally 4 and 8. Then 5 and 9. So in this way, you can see, so here is the 5 and 9. So in this way, the structure is formed for alpha helix. Whenever alpha helix is formed like this. So what it is there? So 1 and 4th, 1 and 5th, 2 and 6, 3 and 7th. So in this way, it keeps on going and it is forming the alpha helix. So if you just go and visualize any protein structure, you take in PDB or in anywhere, and if you see this kind of alpha helix, you can understand that what is the hydrogen bond and interactions between which and which residues. This is after every 4 residues or I plus 4. So if you can remember that it is within the interaction between ith residue and I plus 4th residue. So if we can keep on counting, and this is the hydrogen bond, and this is how the structure is formed. So this is the right-handed coil for 4 to 40 amino acid residues, and it is held together by hydrogen bonds between CO and NH groups of every 4 after every 4 residues. After every 4 residues, and each complete turn of the helix spans 3.6 residues, and it defines the coil's thickness and turn length. Defining the coil's thickness and turn length. So this stability is dependent on correct steric configuration. So this stability depend on correct steric configuration. So the large tryptophan, tyrosine, or small glycine destabilize the helix. Okay, so this stability of alpha helix is quite very important to be determined by the correct steric configuration. And when we have large tryptophan, tyrosine, or small glycine R groups, it destabilizes the helix. So where we need to be that those amino acids don't favor the formation of alpha helices. Proline destabilizes due to its irregular geometry and inability to participate in hydrogen bond. Okay, because it does not have any free end. Okay, so that's why. So just remember which of the amino acids. You don't have to remember. You have to understand and apply the basic concept that in this kind of helix, the stabilization or the stability is quite important. And if we have the large amino acids or small R groups, it will definitely destabilize the helix. Or if we have the proline, then it will also be destabilizing due to its irregular geometry and inability to participate in the hydrogen bonding. The overall dipole moment of the helix caused by individual CO group dipoles affects stability. Stable alpha helices often end with a charged amino acid to neutralize the dipole moment. Okay. So this was all about the alpha helices. So do you have any question or any doubt regarding alpha helices? Anyone? So you have clearly understood that I and I plus 4 residue is what will lead you to the conformation, and it is obviously there will be some amino acids that will destabilize the helix structure, like it proline, like it glycine, because of very small nature, and then of course we have large like tryptophan and like tryptophan, which will destabilize the helix. So now we will be looking into one other secondary that the proteins generally formed are beta sheets. So beta strands are nearly your linear elements of the secondary structure to hydrogen bonding. So you can see there is a one linear strand of the polypeptides, another strand, and they are taking the hydrogen bond. Forming a beta sheet can be separated in the primary sequence by long segments of amino acid and not part of the sheet. In parallel beta sheets, hydrogen bonds are slanted in alternate directions, and the sheet exhibits a right-handed twist. This contrasts with the antiparallel sheets where H bonds are perpendicular to the strands. So you can understand the difference between parallel strands and anti-parallel strands. In the parallel beta sheets, what happens is that are slanted in alternate directions. So you can see slanted in alternate directions, and here they are in the anti-parallel at the perpendicular to the strands. And mixed beta sheet a mixture of both parallel and anti-parallel hydrogen bonding. So this is what comes across the beta sheet. So beta sheet will have your residues and I will have interconnected hydrogen bond. that can be either slanted or can be perpendicular to each other on the basis of the parallel and anti-parallel strand. But in the helix, it will be within that residue and this loop. But in the beta strand, there will be uh, elements and chains of amino acids, and then finally it will be getting matched. Okay. So now after that, we comes under the tertiary structure of protein. So once the secondary structure of proteins are formed in alpha helix and our beta sheet, so now what we are going to do is that we are going to try to understand and identify the protein tertiary structure. So tertiary structure is the 3D configuration of a protein to interaction between residues that may be distant. So the 3D conformation comes after we get the tertiary structure of proteins. So these interactions involve various bonds and forces between R groups and the backbone. So types of bonds and interactions. So tertiary structures are stabilized by hydrogen bonds, van der Waals interactions. So in the secondary uh, thing, you can see that the bonds that it was formed by the hydrogen bonds. So now the bonds that are help to regain its or stabilize the structures are generally hydrogen bonds, hydrophobic interactions, van der Waals interactions, disulfide bridges, ionic bonds like this. Hydrophobic core formation, non-polar amino acid cluster at the core of the protein to avoid water. So this is very important. So if a question comes like this in concept, you can understand. So just think after uh, uh, completing this week one that the different types of amino acids are being made not just to increase the diversity, or it was not just by the nature that nature had tried something and have got the different diversity, but each one of those amino acids, of those 20 amino acids, has its own unique role. The role to make the protein structure stabilize. Some proteins they give uh, extra, what I can say, that rigidity, or that it does not break, like the heat shock proteins or those kind of nature. These 20 amino acids are only on the basis of their arrangements are able to give them the structural stability to form the biological activities, to perform different biological activities, so that they remain together, they do not get breakdown, and they can perform each and every activity very clearly. So this is where it comes very important, the role of each and every amino acid. And the non-polar amino acids, these are clustering at the core of the protein to avoid water, and it is stabilized by van der Waals forces, hydrogen bonds, ionic interactions between polar charged amino acids to contribute the overall structure. Then we have the role of primary structure that we have seen antisense experiment. Then we have the folding intermediates, chaperones, which reaching their stable configuration, and the folding defense you can understand. The misfolding can result in your various orders like disorders and cellular deaths. And finally, the quaternary structure formation is between the two subunits that like the hemoglobins A and B subunit, which forms a quaternary structure. But still, the tertiary structure is very much important. And one thing is that the protein folding is very much sorry. So the protein, so just a minute, yeah. So the proteins foldings are very much crucial in having the 3D structure, and they are very much evolutionary conserved as well. The 3D structure of the proteins are pretty much evolutionary conserved as well. That helps us in getting the data. That is, they help us in getting that particular structure and the biological activity to finally get it captured. So finally, we have the individual subunit structure. We have the individual, each subunit has its own primary, secondary, and the tertiary structure. And then we have the forces that are holding the subunits together by hydrogen bonds and van der Waals forces, specific arrangement and impact of alteration. Okay. So you can understand that it is quite important to have, otherwise it will not lead to the proper function and proper structure and overall proper function of a protein. Okay, so the forces that are holding subunits together are quite important. Individual subunit structure and those structure formation. Yeah, sorry. So protein folding, if I talk about in just a brief, so this is a process. Linear polypeptide transforms into its functional three-dimensional structure. I think all of you are aware of it. So protein folding is basically nothing but the linear polypeptide chain that we get. It's to be converted into a linear polypeptide chain and is transformed into its functional three-dimensional structure. And initial and final states will be that the proteins initially exist as unfolded polypeptides or random coils after translation, and they acquire a stable three-dimensional structure known as the native state. Then we have the information contained in the primary structure, role of chaperones, and evolutionary conservation, as I mentioned, and the importance of correct folding and the ionic interactions and the disulfide bridges. Okay, so this we will find in quite in the reducing. So you can see that in the reducing environment, you won't find it, but where you need to provide high stability and all, there we will be finding. So now there is another concept of Levinthal's paradox and energy landscapes. So will any one of you would like to just give a brief idea of Levinthal's paradox? So you have gone through this in the video, right? So have you gone through this in the video? Huh? So what is Levinthal's paradox or the energy landscapes related to protein folding? Just try out. So any reaction to happen should be thermodynamically stable or favorable, right? Do you agree with my statement or not? Is there anyone who does not agree to my statement that any reaction to happen, it should be thermodynamically favorable? Agree, right? So what we are trying in the Levinthal's paradox and the energy landscapes is that this thought experiment highlights your problem of finding your stable energy configuration for proteins, where an exhaustive search through all possible conformations would be impracticably time consuming. So what is that? If we try to find out the finding the stable energy configuration for our proteins, so exhaustive search through all possible conformations would be very much impractically time consuming. So what it is there? So protein folding efficiency, despite the vast number of potential conformations, proteins fold quickly into their native states, even in complex structures. So, despite the vast number of potential conformations, proteins fold quickly into their native states, even in their complex structures. So, the rapid folding process suggests that protein transitions are guided towards stable states through an uneven energy landscape rather than through random search. Okay, so their rapid folding process should be that the transitions are guided towards stable states through an uneven landscape and rather than through random search. Okay, so this is basically to understand that if you try to find out the possible states, so it should be guided that which will be the favorable and should not be too much random. Otherwise, it will be in a paradox that it will be not possible to find a stable energy configuration. So for each configuration, we compute so QR and FR, then average and repeated for 0 and which is less than the 1. Okay. So molecular chaperones are those which are conserved proteins that assist in the folding. So these are that plays a very huge role when there is for the assist in the folding of the newly synthesized proteins and preventing misfolding under normal conditions and stress such as high temperatures. So any change in the chaperones would be also very much crucial and can be taken. Then assistance during stress, that the chaperones help maintain protein stability and proper folding even when cells are exposed to stressful conditions. And significant advances have been made in understanding the ATP-dependent mechanism used by HSP70 and chaperone families of chaperones. And then cooperative folding, HSP70 and chaperones can work together to facilitate the correct folding of new polypeptide chains. Okay. So as I mentioned, so I'd like to end towards this today's thing. So there is the omics technology. So as we have started with, I would like to finish it there only. So we have understood the basic principles of chemistry, protein chemistry, amino acids, and all. Where we are going to take it forward for understanding towards the entire set of the proteins present in an individual. So there are different omics technologies. So just if I give you a brief, that it is not today, or that certainly we have come across this idea that if we can understand the entire set of the proteins, we can do something very groundbreaking, and we can come across to certain extent. So it all started when scientists thought that if we understand the genetic sequence or the DNA sequence of an individual, then our lot of problem can be solved, and we can easily come and can understand and can tackle many diseases. So it started all in 90s, and in 2000s we could complete the Human Genome Project, but it was a kind of shock for the scientists because it was found out that the sequences are quite similar in nature. The DNA sequences are quite similar, and there are not much changes between the individuals. So then came the picture of other omics technologies, specifically the field of omics, proteomics, which stated that the after the gene or the DNA sequence, it is the proteins that are the plays the roles of particular functional and the structural role in an individual. So this understanding of this is will be quite much useful. Then comes the transcriptomics, where the gene expression that is quite directly or indirectly linked with the protein expression and the proteins. And then finally, now the emerging field of metabolomics, where the metabolites or the functional level of the small molecules are also come into playing into picture, that which also uh, is going you to understand the phenotypic effect at a very later level. At a very level, you can understand the metabolic disorders that are being in place completely miss today's date. So we all can understand at each layer it, and whatever layer we add, we keep on adding the complexity, genome, epigenome, transcriptome, proteome, metabolome. But ultimately, this gives us a picture of all the curated things and in a precision format, and very much important required. So these are the omics technologies. I think you will be going to the omics based technologies among the proteomics. So you have these course. We will be learning introduction to proteomics in quite detail. So you can understand the proteins and its role, its place, and its functions. And then definitely you can explore other techniques as well. As well, in this semester only, I think there is another MOOC course ongoing, that is the proteogenomics, which combines the two technologies, proteomics and genomics, to gain a much comprehensive information. Yeah, so if you have any question related to this week's doubt or today's this live session or anything, you are free to do so. So please let me know that if you have any doubt, any questions. All right. So everything is clear. Okay. So then I think we can. Yeah. So I think we can then end today's class, and I think we can meet again in the next week. So hopefully, as I have mentioned, that you can go ahead and ask different search, like specifically the question that I said, like the trypsin, how it works, what reaction it breaks down. Okay, because this all will be coming in the consequent weeks, and we can look into it. Okay, so I think then we can end today's class, and if you have any